record, no? Okay. Okay,
yes,
thank you.
You can start.
Thank you. Thank you, Luis. First, thank you to invite to develop this e-seminar in the project fruit in the network FruitCREWS. Sorry. First of all, the first part of the presentation is developed to my colleague Deborah Franco. For the reason, I have my presentation, and when Deborah want, it's only say to me to pass the next slide. Thank you, Deborah. When do you want?
Okay, I'm ready, Javier. So when you want, you can pass the slide, please.
Yes.
Yeah. Before starting, thank you very much for the introduction, Luis, and for the invitation to share with you all one of the projects we are working on right now. And this project is REGAVID, and it means literally irrigation of the vineyard. REGAVID project is a Galician operational group, which objective is the development of an irrigation management tool to determine present irrigation requirements and estimate future needs. The partners in the project are Maret Viticultura, as the company I'm representing here. We are leading the project and in charge of prediction models development and software development too. The University of Santiago de Compostela, represented here by Javier Cancela, who's going to explain their work in just 1 or 2 minutes. And we also count with the winery Martin Kodaks that hosts the project with 1 experimental vineyard. And last but not least, FEUGA, that's the entity that is in charge of the relationships with the administration and the dissemination of the project. So REGAVID started in 2022. We started with the election of the experimental plot and the definition of the irrigation treatments. And we selected 2 rows in Martin Kodak's experimental plot to perform 2 different irrigation treatments. So as you can see in this slide, row 8 was selected as the cellar irrigation treatment. It means that it's the irrigation that the seller could use if there were no project. And we have another row 3 in the slide. This row in the first year was a non-irrigation area, and in years 2 and 3 it's the irrigation according to the project calculations. We call it the REGAVID irrigation. So last year we performed the calculations to send the winery staff every Monday a schedule of the weekly irrigation for the following week, with the number of minutes per week they had to switch the irrigation system on according to the data we had gathered the previous week. And now to start explaining how we do this, I'm giving the floor to my colleague Javier. So Javier, when you want.
Okay, thank you, Deborah. Now, previously to start with our main work involved in this operational group, only brief comments about why it's necessary to irrigate in Galicia. Galicia is an Atlantic area with a temperate climate, but in this case, we need, due to the climate change conditions that involve to all parts of the world, we need to introduce some specific model or tools or something to advise the vineyards, seller in this case, if Martin Kodas as member of the operational group, to could determine how is the best moment or how is the quantity of water is required to obtain a sustainable production. No? We have here in the next slide, in this figure, a work developed by several colleagues in University of Baleares. Then you can see the past years. Then it's possible to see in blue colors what is the water balance between our area. Is located in the green circle. We have a positive balance about 600 millimeters in the previous years. Okay, this is the normal situation that all people have in their mind. But when we advance to the prediction to the next year, we obtain this very, very different image. And we can see a lot of areas in red colors. In our case, in Galicia and in the north of Portugal, in other parts of the Iberian Peninsula, this is obviously on Italy and south of France, we obtain negative values in a water balance. This water balance is obtained between the difference into reference evapotranspiration and precipitation during the crop growing season. For the reason, I think so that this image is very clear to explain what is necessary or develop this type of tools. No. Well, the first part of our study was to determine how is the soil variability in the little plot, about 1 hectare more or less, when we developed our study. We used the traditional EM38 dual, this equipment that have the capability to determine the soil apparent electrical conductivity. You can see in both images, big images, the results of the variability related with the first meter, more or less, 0.75 meters of depth. And in the right image, we can see the lower variability in our plot related with the values more focused in the 0.75 to 1.5 meters. The yellow lines show where are established our experimental, as explained before by Deborah. We have two different treatments, but really this second year in 2023, we introduce a third study that is another treatment that is Rainfed to maintain and compare the situation between two different strategies of irrigation when we apply irrigation, that is the traditional in our area. Well, sorry, only to comment. They have about 30 millisiemens per meter of soil electrical conductivity in the horizontal, in the first meters of depth, and in the above, in the below depths, we have depth, we have about 1980 more or less millimeters per second. We have a different values of the soil apparent electrical conductivity. I think so that you know that the soil apparent electrical conductivity is an integrated variable that introduces the texture of soil, salinity, water content capacity, and other aspects as texture or organic matter. For the reason, always before to start experimental SI, we always develop this type of studies to try to reduce the soil variability when we analyze our results at the end. This is the traditional equipment that more of you know perfectly. We take measures with a colander chamber pressure. We have other parameters in the plant, that is a porometer, a chlorophyll concentration meter, or something related with the temperature of the leaves, with a thermal camera. And we have always a TDR 100 to determine the values, the real values of soil water content, and compare with the sensor that were introduced in our project. That, as you can see in our case, we selected Watermark, that is a soil water potential sensor that all people know probably. More of the people maybe that think about the negative results that Watermark could develop in a dry area. But in our case, we have conditions that permit use this type of sensor in a correct way and obtain values, good values to could manage the irrigation. Now in this slide, you can see the results relative to the comparison between soil water content and soil water potential, the traditional comparison. We think that it's necessary previously to pass the information between the sensors of watermark and the real soil water content, developed something similar to this. It's something traditional, obviously, but more sometimes the people forget it and don't develop this type of studies. We have instant measures of the TDR, with the TDR, in this case, obtaining soil water content. This is in the axis X. And in the another axis, we have the soil water potential expressed in centibar in this case. If the people know what is the limits of the soil water matter, the soil water potential sensor watermark that we use, the limit established by the manufacturer is 237 centibar, more or less. In our case, all measures are below these values. For the reason we have a good performance of this type of sensor in our case. The correlation are not perfect. We have about 0.7, 0.8, more or less, it depends. We divide the data in the different treatments to determine if there are some bias or not. When we use different treatments in the different soil, the soil is more or less the same, as explained before. Really, we have two images very, very different and very strange, maybe for you. That is in the left side, we have a comparison between the values of the first 15 centimeters in relation to the soil water potential with the values of the soil water content referred to 45 centimeters. We have a very, very good relation. We have periods of humectation very fast, link it with the irrigation, with surface drip irrigation, sorry. Very high, very strong precipitation, storm with a higher quantity of water in several parts of the following season. And here, more or less, we have obtained the same values, but we have some deviations. These points strange that we are strange and very distance of the curve that we obtain. For the reason we have experiment and working about this to think what is the best relation to modify and introduce this correlation to transform the values of the soil water potential to the traditional soil water content that is more easy to understand, normal to the wine grower. Now in the next slide, you can see the dynamic of both parameters. In the first image, we have the dynamic of the soil water content obtained with the TDR 100. You can see that we have more or less field capacity, more or less about 30%, 31, 32, and the permanent wilting point is less than 10%. But in the first months of the past year, in 2023, we take saturation conditions because we take several instant measures, after a very, very high event of rainfall. For the reason we obtain these values about 40% of soil water content. Then you can see that when it start the months of March, April, or May, there was a lower values in the soil water content. Here we increase the soil water content mainly due to the high precipitation that we obtain in our climate, in the Atlantic area, in the temperate climate. More or less in this part, we obtain a separation between both treatments due to the REGAVID treatment that is linked with our proposal and our project. Start to irrigate with something that how explained previously Deborah, and not start to the irrigation in the cellar. No, for the reason we obtain a difference in the months of June, but higher in the month of July. No? At the end of the cycle, because the harvest was more or less about, the vintage was about 20 of September, we obtain similar points, similar content values due to the rainfall was more or less the same. You can see in the next, in the second image, the dynamic in the same time, the same points or the same date, sorry, to both treatments, obtained with the soil water potential located at 45 centimeters of depth. You can see more or less the similar dynamic, obviously in a opposite direction, because when we have a lot of water, we have lower potential, as all people know. And more or less, we can obtain similar values for the reason and to obtain a good correlation between both parameters. Here we introduce only to illustrate our data dynamic of the different treatments. Sorry for the size, because I prefer to put a higher image. The blue line is the same treatment, REGAVID treatment. Excuse me. The REGAVID treatment is the gray line. Sorry, I have a mistake here with the colors. Gray line is REGAVID. For this reason, obtain the lower stem water potential values. More or less the same, the rain-fed treatment, that in this case is blue, and Martin Koda's treatment, that is the cellar treatment, that only applied 2 irrigation events during the past year, only more or less move about the same values. Now, when our work actually are working about the calibration, now really about the validation of the single KC model to our conditions, starting with the KCB values to the mid-season and late season, and obviously development season, using the Allen and Pereira approach. The past years, we applied 2 different coefficients between pre and post veration. A KCB mid about 0.43 and a KCBN about 0.34, sorry, and a stress coefficient that is the multiplication directly of both values of 0.25. As explained before by Deborah, we apply an irrigation schedule using the ET0 and precipitation predicted to the next week, and then compare and correct how Deborah explained in the next slides. I think so that is all my part, but maybe that Deborah now explain a little aspect more interesting that is related with the estimation of the predictions that is not traditional. This is some of the key points of our group, our project. Thank you.
Thank you, Javier. I resume from here, and before resuming my presentation, I would like to remind that the objective of the project is to estimate future irrigation needs. So keeping that in mind, the first model we have developed is a 7-day prediction model for the ET0 accumulated in that 7 days. What do we do? To develop this model, we have started using the measurements obtained in the first year, both in the non-irrigation area and the cellar irrigation area. Besides, we use real ET0 data measures obtained by weather stations located in the vineyard itself, and even weather stations located close to the vineyard. From there, we have developed artificial intelligence models that allow us to predict the accumulated ET0 for the next 7 days. And at the end of these 7 days, we have the actual ET0 of the previous week. So we can run a graph like this, comparing the predicted ET0 and the real one, the actual one, measured 7 days after. So this is exactly what I'm showing in this graph, comparing this ET0 predicted and actual ET0. And we can see the fit is fair enough. In the worst case, we have a 10 millimeter gap, but let's keep in mind that it's these 10 millimeters are shared in the 7 days. So it's an accumulated 7 days prediction. And you can pass the slide, please, Javier. So as we want to get the future irrigation requirements, beside this ET0 that I repeat again in this slide in the left above, we also need a 7-day rain forecast, as Javier has explained before. So here in this slide, we can see in the image above the ET0 again. To obtain the ETC, this graph would have to be compensated with the adjusted value of KC that Javier told us a minute ago. And in the lower left graph, we see the 7-day rainfall forecast. This graph compares the prediction obtained for the accumulated 7 next days with the actual rain, which 7 days later we know has fallen. So we can compare the prediction with the real rain that has actually fallen in the 7 previous days. And now we are ready to calculate the irrigation requirements that you can see in the graph on the right. We see 3 critical peaks in June 5th and June 6th at the beginning of the graph, and another one in August 30th. These peaks are due to the difference between the predicted rainfall and the actual rainfall. But there is something interesting. These rain events are precisely when irrigation requirements are negative. So there is no need for irrigation. So we can conclude that those moments in which the model performs worse are those moments in which the model is not necessary at all because there are no irrigation needs. You can pass the slide. Okay. And now, with all these models, we have developed a calculator to obtain the minutes per week that the irrigation system has to be switched on. Last year, 2023, the first irrigation week was the first week of July, the 3rd of July, exactly. And in this graph, I am showing you the calculated weekly requirements compared with the real requirements that we calculate a week after. So for the first day, we have calculated requirements of 750 minutes. We sent these numbers to the seller and they decided we want to apply this irrigation in 5 days or in 2 days or in 3 days, but they have to switch on the irrigation system for this period of time. What happened the following week? We calculate the actual requirements for the previous week, and the number we obtained was 780. So there is a 30-minute gap between the predicted requirements and the actual needs. So this correction that you can see there above in this bar, with this purple bar, this correction was applied the following week. And this is the protocol we have used the whole year. we can see that the requirements predicted are very close to the ones that we could calculate a week after. And the last slide, I'm going to show you a beta version of the irrigation management tool that is one of the goals of the project that will be fully deployed this year, maybe in September, October of 2024. And what's the idea behind this tool? The idea is that the user could have real-time information about the irrigation requirements in the vineyard. So in the graph on the left, we can see the soil water content read by the soil sensors. You can see, if you remember the graph that Javier showed us before, it follows the same trend, but let's keep in mind that this graph, it's a beta version, and it was initially, the parameters were initially established in a theoretical way. So it's going to be a work for this year to set this tool with the real values that we obtain for field measurements. And the graph on the right shows the balance, taking into account the rain, the ETC, and the irrigation applied. As I told you before, it's a beta version, but it's the way to start this development of this tool. And that's all for me. Javier, you can pass the slide if you want. Thank you very much for your time. And if you have any question, we'll be glad to try to answer it. Thank you.
Thank you.
We will do the questions after the second presentation. Okay, if you want, you can put the signal, this signal of the hand, or to do the questions after, in order. The second presentation, or the second speaker this afternoon, the place that you are. Daniel, if you want to put your presentation, the screen. Daniel Revillini is the second speaker. In 2012, he received his master's degree in the University of York in UK in environmental management. He received his PhD in 2018 in Northern Arizona University in US to research the plant microbial interaction across multiple study systems, including environmental change across experimental template grassland at Biocon in Minnesota. He did a postdoc in the University of Miami in 2019, and now Daniel is a soil ecology working a Marie Curie postdoc in the Instituto de Recursos Naturales y Agrobiología de Sevilla in Spain. The title of his presentation is FunVine, Harnessing the Soil Microbiome to Promote Sustainable Vineyard Management Under Drought. Okay, Daniel, if you want.
Okay, and is it on your screen? You can see it? It's shared, the presentation?
Yes, it's perfect.
Oh, okay, just making sure. Okay, thanks for having me. So yeah, I am working with soil microbiomes and grapevines. So I'll just get started talking about my project in general. But first I wanted to thank Melissa Venturi, who helped me with some field work when she was here, but also we both thought this was kind of an interesting way to bridge a gap between management and crop science and some of the work that we do with soil microbiomes and soil management potentially going forward in some of these agricultural systems. So just brief summary of what I'm going to talk about, because I'm kind of a little bit of a different researcher for FruitCREWS. I'm going to do a lot of background in what I mean by the agricultural microbiome, some of the relationships between the microbiome and water and drought in particular, ways that we might be able to merge sustainable management practices with microbiome or soil microbiome and the management of it. I'll introduce an experiment we called, it's part of my Marie Curie postdoc, we called it FunVine, so functional grapevine microbiome. And so that's a growth experiment with some very preliminary results, but I will talk about some future goals as well. So yeah, I guess the first question is basically what is the agricultural microbiome and how does the microbiome work in an agricultural setting? Just the very basics. I'm talking mostly about fungi and bacteria. Of course, there's like viruses and phages, but it gets complex. So for simplicity, I'll just be talking almost entirely about fungi and bacteria or archaea in soil. But importantly, there's a microbiome associated with basically every part of a plant. So in soils, in roots, on leaves, on fruits, in fruits, and then even insects that interact with, for example, fruit trees or tomatoes in this figures picture. Insects have their own microbiome, which then leads to a different set of interactions between the plant microbiome. Another thing that's important is like the microbiome is very different at different times of the growing season. So even like pre-germination, germination phases, harvest time, and all the way to death, there's a transition of the microbiome throughout. So timing is also important to understand. And then, so the microbiome, which we would consider, taking a community approach, we would say that these are shaped by their environment. And by that, I mean, it can be shaped by interactions with the types of plants that they're interacting with, the types of insects or other microorganisms in soil. And then, of course, in the case of, for instance, soil, abiotic effects like soil nitrogen, soil pH, soil water content, things like that will change the way that the microbiome composition changes. In the agricultural context, of course, fertilizers will change the dynamics. Things like water management and tillage, like actual disturbance of the soil, can alter the way that the microbiome composition changes. So that's composition, and then when we start talking about function, it's also important to remember that, of course, there are lots of plant pathogens that are fungal or bacterial, but what I typically study are the good microorganisms, beneficial generally to plant growth or plant health or plant nutrition, but as everything keeps getting more complex, there's also a range that these things, it's kind of a scale that the good and the bad interact on. Not things aren't always good and things aren't always bad in the case of plant growth. So moving on with the complexity, I'll just try and bring up some of the functions or roles of the microbiome in the context of plant microbial interaction. So in the figure here, it's important to remember that plants develop very specific, plant species or plant varieties develop very specific microbial communities. And so these things can regulate plant responses specifically, like plant responses, physical responses to drought, physical responses to high temperatures or changes in nutrient content. So in that case, they're dealing with water retention, drought or heat tolerance functions, and then, of course, transfer of nutrients, which is really common for things like mycorrhizal fungi, which associate with most crop plants, including grapevines, and free-living bacteria, or nitrogen-fixing bacteria. So importantly, these microbial communities that, again, are plant-specific, also regulate the interaction with other organisms. So think of this as like they can set up a wall to suppress negative, harmful, pathogenic bacteria, fungi in soil, or for example, insects or nematodes but they can also recruit or encourage other beneficial microorganisms to come from either the soil or, in the case of maybe the leaf microbiome, from the atmosphere. So there's kind of this lots of regulating functions going on between the plants and the microbes. So then to get more specifically to kind of drought tolerance and how the microbiome can promote drought tolerance, there are indirect effects like the way that microbes alter soil structure, increasing aggregate size, increasing microaggregates, and then even the release of these things called EPS, extracellular polymeric substances, which are basically like kind of like a glue in between soil aggregates, which can trap water and increase soil water content just by the presence of potentially more microbial biomass or different types of microbes that might promote this EPS production more than others. They also form things called biofilms, which is mostly bacterial, kind of think of it as a mat of bacteria that might surround a root and kind of have more water as well in a biofilm. And then there are direct effects. So things like arbuscular mycorrhizal fungi, which in this figure here show in the bottom that they, let's see, explore the soil. They go into the plant cortex, so in the plant root, and within the plant root, they actually transfer water directly from further out in the soil than a root can explore, or into smaller pore spaces than a root can explore. So there's the direct water allocation from places that plant roots simply can't reach. And then they also do very impressive things called inducing plant, well, they call it defenses, but it's usually hormonal plant responses to things like drought or pathogen attack. So I'll talk a little bit more about that. So there's this really nice study I just actually found recently where they inoculated, I'll kind of walk through this figure on the top. So SPMX is basically the bacterial inoculants. It's a mixture, 4-species mixture of different bacteria known to promote plant growth. So they inoculated plants, they had uninoculated plants, and then they performed a drought stress treatment for 7 or 14 days. And basically what they found is that only when they were inoculated with this microbial mixture would this abscisic acid biosynthesis pathway express more of these genes, and this is actually, I'm not sure how familiar people are, but basically controls stomatal closure, so it promotes drought tolerance in times of like long-term drought stress or strong drought stress, and so it prevents too much transpiration. So basically, they found that under stress with these microbes added to I believe they were studying rice, they were able to measure specific functional changes with genomics to determine that the microbiome can increase plant drought tolerance. And then there's a whole other set of genes that are constantly being discovered. So we already knew a lot of plant genes that are associated with water transfer, like directly through roots. But now they're identifying fungal water transport genes, and it's been proposed that there's even a form of control between water, phosphorus, and plant carbon to the fungi, where the amount of carbon being transferred to the fungus will regulate the amount of phosphorus or water it will transfer to a plant. So again, very complex, but potentially very beneficial for plant productivity and plant water content, especially during drought. So with all that, I'm going to move to the grapevine microbiome, and basically we know in general, who is there, as in I've identified a lot of the community members associated commonly with grapevine microbiome in the soil or on the leaves. What we don't know is specifically what functions they're performing and how they might respond to different stresses. So of the few studies that I've been able to find that actually test specific effects of microbes on pathogen suppression specifically, there's a few ways. So they can stimulate plant immunity, again, kind of priming these plant defense systems by the addition of particular microbes, and also antibiosis, which is essentially direct blocking of a pathogen using whatever they have in their arsenal, certain chemicals. Vine microbiomes have been shown to adapt to their local conditions. In this case, this study was a global project where they took vineyard soil and characterized the bacterial and fungal communities. So bacterial communities are on the top, fungal communities are on the bottom, and basically the farther apart the dots are, the more different they are from each other. And so in the case of bacterial communities, you see, I'd say just by eye, a bit more differentiation between the countries where they were sourced from than fungal communities. So you might have more similar fungal communities across the grapevine microbiome, but that's just one study. And of course, you can see that Germany, for example, and Denmark had some very big outliers, as did Australian fungal communities. So we're starting to see that there's some filtering just based on where they were sourced from, what country. And what we're wondering in my project is how, if we can find something similar for function, so microbial functions specifically, not just community members, because we want to start to understand how we can measure the good versus the bad. And so in our case, we're looking at gradients of nutrient limitation, drought stress, or water availability, and then temperature stress, or mean annual temperature, things like that. So across large gradients, how is that shaping the way that either the leaf or the root microbiome functions? And we do this with some molecular metagenomics, essentially, where you look at all of these functional genes that code for specific enzymes. And then more specifically in the growth experiment, we're wondering if using soil inoculation, can we manage for this, what we're calling adapted functionality of the microbiome. So when you source soil from different vineyards that has a different background, environmental background, management background, different grapes, of course, we're going to ask, can you take soil from other places, apply it to a growing system, and observe any physical responses to drought or fertilization, in our case, to promote these beneficial functions. So just briefly, I want to go over some kind of definitions. Sustainable management, these are that I used in my title. Sustainable management, I'm basically saying minimal fertilization or minimizing fertilization, minimizing pesticide use, so minimizing chemical inputs into a system, promoting stress resistance generally through a microbiome or through plant selection, crop selection, variety. And then maximizing output. So of course, economic output, yield, things like that, or biomass productivity is important in any agricultural system. And then the microbiome functions that I'm talking about, again, are increasing nutrient cycling, increasing water retention or tolerance to drought, and increasing pathogen suppression. So can we do these things using soil inoculation? Specifically, we're trying to minimize the negative effects of drought on early vine growth in my experimental project, and identify the optimal sources of the microbiome. So where can we collect soil? from. Can we use a whole soil inoculum to increase some of these beneficial functions that actually end up leading to higher, better plant growth or yield? So again, I have very initial results from my project, but bear with me. But I do actually have some biomass responses, so that'll be nice. And I'll have some predictions for where I think this might go when we get more data. So you'll notice immediately that this is a little different than some of your field experiments, especially with grapevines or other fruit trees. But nonetheless, it took a lot of work, so I just wanted to thank a lot of the people. I had so many people in our lab help, mostly just moving soil, digging soil and moving it around, which took a very long time, lots of work. And then finally, we just set up, as you can see on the right, an irrigation system, essentially. We just had drip irrigation across all of the plants, and I'll give you some specifics about what we were growing. So we had 30 sources of soil inoculum, so from all over the world. I'll get into more detail on that. It was only 25 grams, so in 18-liter pots, that's not too much, but we did observe some effects, which is nice. We had 2 grapevine varieties, Garnacha and Merlot, and they were of the same rootstock. 2 fertilization treatments, which was no fertilization or kind of a moderate level of fertilization. 2 water treatments, which was no drought, consistent watering, or a pulse 2-week drought towards the end of the growing season. And then basically at the very end of the drought, we harvested all new vine growth, so anything that came from the first year rootstock. So a little bit more detail about the inoculum. We basically, again, had vineyard soil from 8 countries. We had 30 different inoculums, so some countries we had obviously multiple samples from around Spain, for example. I think we had 8 different sources, but all of these maintain a big gradient of mean annual temperature or mean annual precipitation. So a big environmental gradient that formed them. 25 grams per pot, and then we do have a lot of, or we will have a lot of background knowledge of the soil, basically soil chemistry and soil microbiome composition. That is still in the works. And then for watering, basically we were measuring it by liters per pot per week, and then the drought treatment again was actually no water for a total of 18 days. That was when we just didn't want completely dead plants, but close to it. So very long and intense drought treatment. And so again, finally for my very preliminary results, we would say the drought was a success, essentially because on the left you can see the no drought treatment, on the right you can see the mean of the drought treatment, which was about half of the soil. So this is the soil water, percent soil water by mass. So essentially maybe a weird metric for you, but anyway, there was about half of the water in the drought content. So to me this indicates a much drier soil than the ones that were watered consistently. So the rest of the results are basically vine dry biomass. So we just harvested all new growth from the vines and then dried everything. So this is all dry biomass. And what we found essentially was that the variety, Garnacha is on the left and Merlot is on the right. And then for the drought treatments, there was either, again, no drought, consistent watering, or drought, which was the pulse, about 3-week long drought. And ultimately, these are colored by fertilization, but what you can see is there's no real fertilization effect. It was not as we kind of expected, to regulate the growth responses to drought. We were basically pretty surprised that fertilizer had no effect on total biomass in this situation, but there was an effect of the variety where Garnacha had more biomass output, and drought definitely decreased biomass for both Merlot and Garnacha. And then finally, this is vine dry biomass again on the left. These are blocked off by Garnacha on the top, Merlot on the bottom, and then again, no drought or drought, and colored by the source country of the inoculum, so the soil that we added to all of the pots. And yay for me, there's some variation here. I was a little scared because this was kind of a blind, just adding soil to pots and hoping that something happened. Ultimately, we would say that there were some strong effects of drought, and then there were some strong interaction terms with the inoculum source. So for example, these soils from Chile, without drought, promoted really high growth of Garnacha, and then with drought had basically the lowest biomass output. Similarly, it looks like Portugal had the soils sourced from Portugal had some interesting effects where there was really no response to drought under these soils, so biomass didn't change of the vines depending on the drought treatment, where in most cases drought had a severe decline in biomass productivity. Finally, I then looked at the soil source, the environmental gradients of the soil sources, and we found that environmental conditions, in this case mean annual precipitation, had at least a interesting effect in the drought and Garnacha treatment, where you can see as soils from drier climates, which are dark brown on the left, versus soils from wetter climates, which are in blue on the right of each of these panels. In the drought and Garnacha treatment, you can see a slight slope upwards to where the wetter soils actually had higher biomass than soils that were sourced from drier climates. And importantly, I would say that there was no, they basically maintained productivity with or without drought. So the soil inoculum have something going on. Unfortunately, I don't have any of the microbial data to blow your minds and tell you what all of that means, but there's something that happened with the inoculum, so that's where I'm excited. So just going forward, it's important, obviously variety selection will be important. We noticed some differences between Garnacha output versus Merlot, especially under drought. Soil sources affected the way that they responded to drought, Garnacha mainly, and also it was the high mean annual precipitation soil sources. And then what we don't know, unfortunately at the moment, is what conferred these changes in productivity. Was it nutrition based, was it water, or was it pathogen suppression? It's kind of hard to tell at the moment because we don't have the data. But that's kind of where we're going to look in the future is we're going to take all of the soils, do a lot of functional profiling, and then identify gene increases or decreases based on these treatments and plant varieties and soil sources to see what's actually related to these plant biomass responses. I think I have a little, I just want to briefly talk about kind of what I think where this can go in the future, which is actually managing the microbiome to optimize these beneficial functions under ideally the lowest possible fertilization rate. So in the context of drought also, these things will change, but this is all theoretical, but the goal is to maximize productivity maximize beneficial functions, which in my little figure are green and yellow, and minimize negative functions, microbiome functions, which would be some things like oxidative stress or pathogenicity, so pathogen strength. So in the future, we hope to be able to identify kind of the point at which you can maximize productivity and minimize negative effects from the microbiome, and keeping fertilization down, of course. So many thanks to my lab members and everybody that helped, and again, Melissa for inviting me to, or introducing me to FruitCREWS, and yeah, any questions for me?
Okay, thank you for both presentations. We have some questions. The first one is for Javier and Deborah. I think the name is Akin Melkensen. If you want to turn on your microphone and do the question.
Yeah, sure. Hello, everybody. I have a question for the first presentation, actually. I was curious for the prediction, what kind of algorithm have you used? What kind of statistical analysis is this? I think you said something like artificial intelligence, but what's the algorithm?
We are using neural networks.
Neural
so
networks.
Yeah.
Okay.
Basically, we are using neural networks, but I don't have here exactly the number of neurons, the details of the algorithm, I mean.
Okay, and which statistical software did you use to perform the analysis? I'm asking just from curiosity.
Statistical software, I think it's R.
Okay. Yeah.
And MATLAB, I guess. R and
Okay.
MATLAB. Yeah.
Okay, thank you.
You're welcome.
Only to complement, we used to the both other software program that is the Canime and TensorFlow techniques to complement the results.
Mm-hmm.
Okay, the second question is for Daniel. Is the Alpha Thoreau? I'm sorry if I don't say good your name.
Hi, good afternoon. Thank you very much. Very nice presentation. Actually, I have a question for three of them. So I will begin first with Daniel. Just a curiosity, Daniel, because you showed that so when you are talking about inoculum from different countries, you are talking about soil. So you are growing your plant in the soil, okay?
Yes.
Okay. And
from a vineyard.
So, sort of vineyard. So do you have any, did you check the soil water retention of each soil? If they are the same or not? Because
of the inoculum. Of the
different soil, because you show that in Chile, in drought, you have less biomass. And in Portugal, what happened that you have the same biomass. So the first thing that I'm thinking about as like a physiologist is that the water retention, the soil is not the same. Because we used to work with drought in 1 week and we see differences in the biomass. So depending, yeah.
No, that's, so let's see, have we measured water retention of the soil? I'm not sure if we have, but I will now. We have all the soil, so we can measure basically everything. We were gonna, in our proposal, what we were measuring was basically most of the more chemical content, so nutrient content, pH, I don't know if we were going to do microbial respiration, but yeah, water, I don't know if we plan to measure that, but I could pretty easily. We have all of the soil available for.
And another question, when you are doing some drought experiment in pots, are you trying to avoid the soil transpiration or not?
So yeah, it gets a little confusing, obviously, growing anything in pots, grapevines especially. But yeah, for soil transpiration, there was nothing we did to regulate transpiration. So it was really just if the drip line was on or off, and it was a very long-term drought, right? I mean, it was 18 days with no water for half of the pots. Yeah, there was no regulation of transpiration though. So again, not sure how that could have affected either, the water, actual water content at the end of the drought, or so different worlds. These are things that I wish I thought about, ahead of time with the plant physiologists. So
yeah, I was curious about this. I think that you maybe have to check this and to be sure when you compare the different soils, at least.
Of course, yeah.
So,
thank you very much.
Thank you very much, Daniel. Okay, now a curiosity to Deborah and Javier, because as I was a little bit confused, maybe I didn't get it. You show it. So when you are talking about the ETC, which percentage of ETC are you irrigating? 100%. So the irrigation program is calculating for 100%.
No, we introduce KCB traditional that starting with the values compilated by the bibliography that I have showed previously, the KCB, and then multiply for a stress coefficient, in our case, 0.25, for the reason we aren't irrigating, as you are asking, a full irrigation. No, really, we have a deficit irrigation treatment.
Yeah, I was curious because I didn't get it at the beginning. And then Javier, you show a graphic. It's very about the comparison between REGAVID and Martin Codex. Yeah. And you show that REGAVID is irrigating more, so is applying more water in July, August, and September.
Yes. Really.
So just a question. So my question is, if you are irrigating more, are you getting something more in terms of quality or quantity or vigor for the next year? So
really, we have a higher production. That is not the main objective of this study. But it's only to compare the effects, not only in production. We have also information on volatile composition of the grapes to determine in a wide variety, in our case is Godello variety, if we have a difference or not when we apply a strategic management through our tool REGAVID, or using the conventional rules that the Martin Codex use traditionally.
Okay. So you are not
exactly the.
Okay, you are not considering the phenology also.
The phenology.
Yeah.
I talk. We talk about the option to apply different crop coefficients when previously to better song or not, and really when we have the necessity to apply water when our model define it in base of the prediction or something like that.
Yeah. I was not very concentrated in this moment.
No problem. Okay. Thank you, Alpha.
Thank you, Javier. Thank you, Deborah. Thank you.
Okay, I see that there are other question in the chat, but Daniel answered. But I do the question, and maybe you can answer here. Did you look at the nutrient composition of the soil you use it? Is possible that there were enough nutrients to start with, and that is why you didn't see an effect of the fertilization?
Yeah, and I answered in the chat, but yeah, just to say, yeah, we will have all of that data. So the background, soil inoculation, nutrient content, basically as much carbon, nitrogen, phosphorus, all of the micronutrients. We're gonna look at everything we added and also the background soil, because it's important. All the plants were grown in the same farm soil, and then the inoculation was only 25 grams of soil on top, like a top dressing. But yeah, we will have all of that data. I'm assuming that the background soil across all of the plants and all of the treatments maybe just had enough nutrients already, and or simply maybe the fertilization rate was too low. I'm not sure, but yeah, we were a little surprised to see no fertilization effect really at all. But that's kind of the way we're thinking at the moment. We just don't have the data just yet.
Okay, we have other question. The Kamut, if you want to on your microphone.
Hi. Thanks for a very interesting presentation. I have a question because you were mentioning about drought, no drought. Usually, you know, when we grow the plants, we well watered, there is an excess of water or there is no water. So basically, my question is how you induce the drought, and is there any quantitative measurement of drought and no drought in your experiment?
Is there any? Sorry, what was at the very end?
Like, how did you measure the how do you measure the drought? Is there any objective measurement of the or quantitative measurement of the drought?
Not really, other than at the end of taking the final soils at the very end. So that's what I was comparing when I used the soil water percent. That was basically percent. It's basically the water mass content of the soil. So I had fresh soil and dry soil, and after drying, I basically said everything that was lost was water. So during the drought, there was just no water addition whatsoever for half of the pots for 18 days. The other pots were well watered throughout. So that's
okay. So and how much percent of soil water content was for well water, and how? So does it mean zero soil water content for drought? Like no for no drought for drought?
Right. So everything was watered equally, the same for let's see, I'm forgetting the number of months, maybe 4 months. Everything was grown together with the same water content, the same irrigation. And then simply we turned the irrigation off for half of the pots for 2 weeks in the end of August. And then let that go for 18 days. So the drought treatment was just essentially a very strong, it was whatever residual water was in the pots was left, but no water was added for 18 days to half of the pots.
Okay, thank you.
Yep.
Okay, Daniel. Hi, Daniel. Javier, perdón, I'm sorry.
Thank you, Luis. Daniel, only one question. I can see that your plots are very near between us. My question is if it's possible that are there some problem related with some interactions between plants, or are you think that not because your experiment is more focused in the soil and not the components in leaves, or I don't know. It's only a curiosity.
No, I mean, it's a very good point because even my advisor, he was like, oh, it seems that just grapevines inherently, when they're close to something, a wall or tree, trellis, for example, they'll put out more growth the second they start to feel, attached to something. So there was no way really for us to control that. It was just about space requirements. But as far as transfer, the only way that we could control any of the soil stuff was not allowing water to splash between pots, so everything was drip irrigation. So there's really no controlling any of the above ground interactions between the plants. So that's the simple answer, is we have no way of knowing.
Thank you.
Thanks.
I think that we don't have more questions. I will do a fast question for Daniel. One for Daniel and another. Well, I know that in fruit trees usually the people use herbicides for reduce the weeds under the trees. Usually the people is using more mulchings. What do you think about the apply herbicides, mulchings, and all these things for increase the microbiome?
Yeah, no, it's a good point. So herbicides, in our case, I mean, we did have every week I would go and literally just hand weed everything, because I had it in pots. It was, it was a lot of pots, but it was pretty easy to just pull the weeds. Obviously, people wouldn't really be doing that on a farm scale or out in the field, but as far as adding additional, like mulch layers or anything, top dressing covers, yeah, I mean, it obviously would have some benefit to water retention, I would imagine. Microbial, yeah, I'm not sure. It could have an effect for sure, but it's just really too hard to say if it would be good or bad. You might introduce pathogens. I don't know. It's always hard to consider what adding something across an entire farm would do.
Okay, thank you. And
Thanks.
Javier, it's enable REGAVID for the growers. Do you have anything? Well, it's the growers can use your one.
Our platform. Yeah, in this case, it's an experimental proposal, obviously, because in the area where we develop our experiment, they have a extreme temperatures during the summer that more of the case they need apply water, irrigation in this case, no, obviously. But they have a problem with the available water during the summer. For the reason, they are very interesting to now the real effects on how and when apply the water. But for example, in the first year in 2022, we have a problem with the available water to could apply our irrigation treatments. Really, they are very, very interesting. And at the end, we will try to put the best tool, simple tool, a friendly tool, to that they could apply during the proximal years, obviously. For the reason they are involved in the project very hard.
Yeah, and I would like to add that the sensors we are using are affordable. So that's the reason why Javier told you before that we were using watermark sensors, because we maybe could use better sensors, but more expensive sensors. So the final goal is to achieve a system that the viticultor or the seller can use, and it's friendly and it's affordable.
Okay, thank you. Melissa, you have a question?
Yes, I have a question for Daniel. Hi, Dan. And I was saying, just because as physiologists, for us it's not so easy. We're not used to maybe to consider the soil microbiome. So in your opinion, there is maybe also something simple or some parameters that we should include in our research when we do drought stress trials? Do you think?
For droughts, yeah. Hi, and let's see. I don't know. Yeah, that's for something simple. It's hard for me to say exactly because I think microbes are so site specific, crop specific. I think soil specific, but it's hard. I don't know if there's some comprehensive, like if you have more fungal biomass in soils, you're going to have more higher water retention. You know, the way I'm thinking, that would make sense. Like fungal biomass could be a sign that you have maybe more moisture or maybe the ability to retain more moisture. But it's really difficult to say like a one or two answer thing. Yeah, it's a new field for me, really focusing on drought specifically. So that also is a little bit not, I wouldn't say out of my field, but I don't know exactly like all of the literature specifically offhand with the microbiome. But potentially something to do with soil aggregate structure also, like soil micro aggregates specifically, because that usually indicates a higher that polysaccharide, the EPS, which is all microbial. Like, so microbes put out these things that are kind of like glue-like, and also hypha, like fungal roots, basically just form a lot more micro aggregates. So there are ways, pretty simple ways to measure micro aggregate percentages in soil. So soil structure maybe could be a way also, but theoretically.
Yeah, still in process, but thank you.
No, thank you.
Okay, I think that we don't have more questions. We can finish the seminar. I want to say thank you. Yeah, Francesco, one question. Sorry.
Yes, I had a question for the I had a question for the first presentation. So for Deborah and colleagues, about the calibration of this artificial intelligence algorithm, because you pointed out that the sensors you use are not expensive sensors, but generally for artificial intelligence algorithms you need lots of data. So I'm wondering what data you needed for calibration, and if you tested about the effects when you change site, because I guess calibration is probably site specific. Thank you.
Deborah.
Yeah. Are you going to talk about the watermark? Yeah.
No, for me, it's very your question. Thank you for your question. Is very interesting. Really, the objective of the use cheaper sensor is only to control and manage the real necessity of irrigate or not. But I think so that maybe that in our presentation we don't focus exactly in the part of the artificial intelligence. The artificial intelligence focuses in the prediction of the ET0 and the precipitation. For this reason, we are very interested in increase the capability of this system to obtain best values, but it's not related with the sensor that you use to control the irrigation or the effects of the irrigation, of how determine when to irrigate in a vineyard in our case. In relation to your second question about the site, in this case, we start with this project, but we have in mind other areas to test the effects of the change the climate data to other part of Spain or another part, and determine the precision of our algorithm to obtain good prediction in a week. No, that is our screen of prediction. I don't know if I respond your question, Francesco.
Yes, thank you.
Okay, we
So
have
nothing left for me to say. Javier explained it all. So thank you.
other question for Daniel in the chat box. What was the medium inside the pot during the experiment? Gap, wine, soil, or medium like feed per liter? Where is the number of type of bacterial population determined in the medium at the experiment start?
Oh yeah, there. So, sorry, I didn't know if I was muted. Yes, so it was just farm soil. So we collected soil. We have an experimental farm at IRNAS here. So we have a farm site. We basically just had a tractor pull up soil from a different part of the farm. We brought it over. We screened out large rocks only. So it was kind of to get rid of very huge rocks to maintain a similar soil size at least, or rock size, aggregate size. But that was it. And so what we did, yes, we will sequence the microbial community. So we'll know what the background soil was, both nutrients and microbiome. So we'll know fungal and bacterial communities at the start. And yeah, that background, the growing medium was, again, for all of the plants had the same growing medium. And then when I'm saying soil inoculum, it was literally we took 20 grams of soil from different sites and put it on the top of that live soil. So we will know that in the future when I actually get the data back. So, thank you.
Okay. No, I think it's all. Well, I want to say, well, if you have any questions, you can contact with the speakers by email. I think that all of them, they put the email in the last slide. I want to say thank you, Daniel, Javier, and Deborah, for your good presentations. I want to say thank you and all attenders or all people that is here for all good interaction that we had, and the FruitCREWS people. Thank you very much, and it's all. Good afternoon.
Thank you.
Thank you, Luis.
Thank you, everybody, and thank you, Luis.
Bye bye.
Bye.
Thank you. Bye.
Thank you all. Bye bye.
Bye.
Thank you. Bye.
Bye bye.